Hello, good afternoon, everyone. Welcome to the session, The Future of Health. This online conference is organized by Goethe-Institut, Indonesian Center for Digital Society and Engaged Media. My name is Safarina Malik, and I will be your moderator for this session. My work is related to lifestyle disease, Nutrigenomics, Nutrigenetics, and microbiome. And I am part of the Indonesian Nutrigenomics and Nutrigenetics Society, Asia Pacific Nutrigenomics and Nutrigenetics Organizations, and the Microbiota Vault. In the next hour, we will listen to and have a chat with our speakers, Herawati Sudoyo from Indonesia and Danuta Gawel from Sweden. Personalized medicine is the future of healthcare. Personalized medicine uses an individual's genetic data to guide treatment of diseases and select proper medications. Its implementation will transform the healthcare sector, but can we really be sure our data is being handled carefully? Should we give our medical history to private organizations or companies? How will vast amounts of genomic data change public health? To get answers of these intriguing questions, let us listen to these two experts. I would like to remind you that if you have questions, you are welcome to type them below in the Q&A session. And we will have time afterwards for Q&A, and we will try our best to answer all of your questions as long as time permits. Our first speaker is Herawati Sudoyo. Herawati is the chairperson of the Medical Commission of the Indonesian Academy of Sciences. Previously, she was the deputy director of the Eijkman Institute for Molecular Biology in Jakarta. So, Herawati, the time is yours. You have 15 minutes, and then I will remind you at 5 minutes before the end of your time, and then 2 minutes before. Thank you. Please, the time is yours.
Thank you very much, Dr. Malik. Good afternoon, everyone. I would like, of course, to thank the Goethe-Institut to invite me to talk in this important webinar. The importance of public health has long been understood, but in the past decade, its connection to the broader healthcare journey and importance for the sustainability of health system has become clearer. Public health should be treated as an integral part of healthcare, embedding prevention and health promotion into people's lives, regardless of whether they are affected by disease, through early detection and screening, follow-up for disease prevention, etc. A data-enabled approach to population health management and risk stratification, also the so-called precision public health, allows to identify those most at risk and customize intervention to specific work group. This is particularly essential given growing amounts of information on people's behaviors, risk factors, and their impact on health. Let me share with you all personal healthcare, the future of health, the future of public health. Dear all, genomic sequencing is rapidly transitioning into clinical practice, and implementation into healthcare system has been supported by substantial government investment, totaling, if I can share with you all, about $4 billion at least in 14 countries. These national genomic medicine initiatives are driving transformative change under real-life conditions, while simultaneously addressing barriers to implementation and gathering evidence for wider adoption. We saw the diversity of approaches and provide a roadmap for sharing strategies, standards, and data internationally to accelerate implementation. Indonesia just launched in 2022 a biomedical and genome science initiative, BGSI, with the aim to sequence 10K individuals from 6 hubs with different topics. couple of years ago, genomic sequencing was restricted to the research environment. Now, it is increasingly used in clinical practice. And I think about over the next 5 years, genomic data from over 60 million patients is expected to be generated within healthcare. Maybe Indonesia already also could becoming one of the country who play an important role in that activity. So what is the issue on the future of public health, personal healthcare? As we are entering the era of genomic and its derivative and big data produced by the finding in this very short time, I would like to try to answering questions such as, how will vast amounts of genomic data change public health? What are the risks and the consequences? What are the ethical challenges of sharing such data? In recent years, a fundamental shift in the way of healthcare is practiced, delivered, funded, and experience has emerged. As the global burden of disease has diversified and technology has become significantly more advanced, both the health needs of individuals and populations are changing. These developments have given rise to personalized health. It is a paradigm shift from a one-size-fits-all treatment of disease to maintaining a high and healthy quality of life by applying the right health intervention for the right person and population in the right place at the right time. Precision medicine, which is informed by each nature, each person's unique clinical, genetic, genomic, environmental adaptation and lifestyle, is emerging and considered to be the future of targeted health prevention, diagnostic, and treatment. Advances in high-throughput sequencing technology increase computational capacity for analyzing large data sets, coupled with steep decline of cost, have revolutionized biomedical research in recent decades. It is not just play alone, but there is integration of whole genome sequencing, genome-wide association study, multi-omics with clinical data. There is a tripartite how personalized healthcare solution could be achieved. Tripartite of system biology, personalized healthcare, and molecular epidemiology. Molecular epidemiology concern of disease risk factor, disease evidence. Personalized healthcare will concern about patient stratification and diagnostic. Both supported the global system biology, in which we will examine the genome and environment. Genome is not only the genome, but and its derivative, transcriptome, proteome, and metabolome. Ladies and gentlemen, the completion of reference human genome sequence has facilitated the discovery and cataloging of variation in that sequence among different individuals. So genome dataset analysis allows us to map out strategy for focusing on specific population. For generation of relevant important tools, and for us to understand the effect of genetic variants on disease susceptibility and drug responses. So understanding how gene expression varies across individuals and environment is fundamental for the success of molecular medicine. So I cited this quote, your genome knows much more about your medical history than you do. Oops. So what is the difference between genomic for research and genomic for health? Research genomic is our ability to answer research study question, whereas healthcare genomics is ability to impact healthcare diagnosis and treatment decision of an individual. Number of genome sequences needed. It's depending on population size of patient with specific criteria for healthcare. And you know that the research genomic will take longer, can be far, far longer. But for healthcare, we need only weeks, if not a month, to depending on disease. So there are, we can see the difference between genomic for research and genomic for health. But regarding the data sharing mechanism, for healthcare genomics, there might be a federated access to secure data virtual, pooling of summary and intermediate statistics. Research genomic, aggregation of anonymous data, and local download and processing. So I would like to share with you here the diagram of how actually disease burden, depending on the cost and also the activity and the approach that we should. Baseline risk can be actually tested or examined using stable genomics. Could be whole genome sequencing, haplotype mapping, or gene sequencing. That is for the aim to assess risk.
Prof. Hera, I'm sorry, you have 5 more minutes.
Yes. So the future of precision medicine in Indonesia is contribution to International Precision Medicine Initiative, bridging gaps with ethnic-specific data. It takes a genome to understand a village. It takes a lot of genome to understand Indonesia, because Indonesia is host of an array of human diversity that remains underrepresented in modern biological survey. We have about 700 languages, and for about 500 ethnic population. So we have data. First of all, we do small activity using 50K SNPs, and now we are adding a lot of whole genome sequencing, starting with Genome Asia 100K project and Indonesian genome diversity. Genome Asia is pilot phase of the Genome Asia 100K project, data set from 1,739 individuals from 64 countries across Asia. And we have catalog of genetic variation, population structure, disease association, and founder effects. What is the result of that data? So although the number is still small, that I presented here, but we already could see prediction of adverse drug response is varied between population. The one in the box was several ethnics from Indonesia, which has responded to several drugs, and we look at the result. Warfarin. Warfarin is an anticoagulant. It treats and prevent blood clots such as DVT or pulmonary embolus. But the use of anticoagulant is actually reduce the risk of stroke or heart attack. this SNP is high, very high percentage in Flores, almost 70% in Mentawai, and 90% in Nias. So by looking at that, you can see actually how they respond, the ethnic population responded to certain drugs. This is also data on CYP2C19 haplotype distribution to look at pharmacogenomic data of Indonesian population from West Indonesia, East Indonesia, and Papua. And we can see it's varied from one to another.
You have 2 more minutes, Prof. Hera, sorry.
It's malaria as a model of selective agent. Also, it is regarding, there is a medical relevance with Genome Asia dataset. We could see the frequency of beta thalassemia variant. What about ethics? Ethics was conducted here is in underrepresented population. There are a lot of discussion why we have to do that, because most of studies were conducted in the population. So what they have, issue, sample collection, and consent process is very, very important regarding working with population in the underrepresented country. So in summary, implementation genomics into healthcare might be different in high income country as compared to low and middle income country. Implement in social and economic context will address areas with major burden to global disease. Broad implementation will be crucial in building representative population reference databases that improve variant interpretation. We should develop strategy and capture experience and engaging cultural diverse indigenous population, public, and also other stakeholders. We should compare national consent procedures, reduce heterogeneity, identify common features to allow global data sharing, explore new models. And the last one, the most important one, international collaboration is needed. Thank you very much, Dr. Malik. And thank you all for your attention.
Thank you for the excellent presentations. Now we will continue to our second speaker, Dr. Danuta Gawel. Danuta is a senior bioinformatician at Mavatar AB. Her work focuses on creating a tool that supports clinicians to personalize medication based on digital twins of patients. What is digital twins? This is something new. Danuta will share with us her knowledge and expertise, and we will listen to her. So the time is yours, Danuta, please.
Thank you very much. So yes, I'm working currently at Mavatar. This is a spin-off company, which means that it has its roots in at the university. It was founded in 2018 in Linköping in Sweden, and is also a part of Swedish Digital Twin Consortium. And our aim in the research and now also in the company is to use digital twins in order to improve what medicine is given to a patient, so that the match between the medicine and the patient is better. So I will start this presentation with the definition of what digital twins are, especially in the context of personalized medicine. I will also briefly mention what is done currently in that field and why do we even need digital twins. I will also mention how digital twins can work in clinic. I will very briefly mention future possibilities and challenges. So what is a digital twin? Well, the most common or basic definition is that the digital twin is a replica, a digital representation of a physical object. It could be a model of a city, it could be a model of an engine, telephone, pretty much anything we can see around us. And we can use those models in order, for instance, to simulate specific events like flooding, so that we can then improve if we have a model of a city and we simulate flooding, we can improve how the city is constructed in order to either avoid the flooding or reduce all of the bad events that comes with it. Now, digital twins in the context of personalized medicine are thought of as models of organs or structures within the body that can be also expanded to incorporate external factors. So here are a few examples of the current study in the field. So we can use digital twins, like here in the example, to model the facial structure in order to provide suggestions for the appropriate orthodontic treatment, or we can use digital twins of heart to simulate the outcome of the interventions so that the clinicians can first use this tool in order to assess and prepare themselves to plan what interventions would be best for the specific patient. Those models can be also used in order to simulate the evolution of the disease and how the drugs would impact specific organs like liver and others. What we are focusing on in the research team that the company started from and also in the company is to create models of specific patients where we incorporate we plan to incorporate a lot of information, genomic, transcriptomic, proteomic, environmental factors, clinical variables, etc., in order to create models of individual patient. So each model here represents this one patient, and those models can be used to test thousands of drugs simultaneously in order to predict which medicine is going to be most efficient to treat that patient. But why do we need digital twins? Well, medicine, according to the Food and Drug Administration report, is inefficient in 40 to 70% of the patients with common diseases. 40 to 70% of the patients with common diseases do not get the relief from the medicine they were prescribed. This is an enormous cost for the system, as you can imagine, when the patient returns to the doctor over and over again for finding new treatment. But this is even most important when we look at the patient's suffering, when the patient doesn't get the relief from the treatment, but might suffer side effects. Digital twins can also help in the context of reducing the cost for identification of the new medicine. When we think of that, the digital twins can help us repurpose the medicines that are already there. They are already tested, they are already in use, but we can find new conditions and new diseases, whether those drugs can also help. So our main aim is to match the right medicine for the patient, and how can we help clinicians to make the right choice. At the moment, we are focusing on the transcriptomics, so how the genes are being expressed in individual cells. The most common computational analysis assess the gene activity in thousands of cells simultaneously, but as a bulk, as a one batch, as a one group. So what we can infer from it is an average representation. So the drugs predicted based on this average representation would be successful only for the most strong signals, for the most common factors. We can compare bulk analysis to as if we would taste a smoothie, a drink that is made of blended fruits. We'd only get to taste one thing. If we have a lot of strawberries inside, we would taste mainly strawberries, but we might not feel blueberries, for instance. Single RNA sequencing, this is a cutting-edge technology that allows us to measure gene activity in individual cells at the same time. So we can get to taste all of the fruits separately instead of having this overview taste of a smoothie. So this is the technology that we are based using in our work, that we are using as a base for our work, where we get to see gene activity in each individual cell, and therefore we have a better resolution in order to predict which medicine will be most efficient. What we do is that we create models representing of how the cells interact in the patient. So here each node represents one cell type: T cell, B cell, dendritic cell, etc. Each edge represents interaction, which means that the cytokine released from this cell might react with the genes inside this cell, for instance. Now, the cell that is in the center, which means that it receives a lot of signal, sends a lot of signal, is then assumed to be most important in that context, because it can distribute and receive a lot of information from the neighbors. At this hypothesis that the central cell is most important, we have validated in several projects which are currently published. And the drug predictions then would focus mainly on the central cell, but not only. We also include information from other cells as well. So the idea is that we can use the genes whose expression is significantly different in a patient compared to a healthy control in order to predict which drug is most probable to change those genes' expression back to the normal levels. So we compare the expression levels in a patient to those that we observe in a healthy control. We map all of the genes that, so to speak, misbehave in a patient as disease genes. And then we identify drugs that are most likely to treat those disease genes in every cell type. This idea, those principles, we have successfully implemented in a mouse study of rheumatoid arthritis, where we have focused on the most central cell type. We have made the drug predictions, and we have identified bezafibrate as one of the best drugs to treat the rheumatoid arthritis mice. And in fact, this drug helped the mice, even though the drug indication is for hypertension and not rheumatoid arthritis. This drug was efficient to treat the mice. But that was in mice. And now what happens in human? This is a study of Crohn's disease, and two individuals are presented. The models for one individual is greatly different from the other one, even though both patients were diagnosed with the same condition, Crohn's disease. One of those patients was indicated as a non-responder to one of the most common treatment for Crohn's disease, namely anti-TNF treatment. And that was also reflected in the predictions of which drugs we would recommend to treat the patient based on the ideas I have presented just now. And those ideas, those principles, we are now aiming to implement so that they can work in clinic, so that we can help clinicians make the decision of which medicine to prescribe to a patient. So how can that be done? Now, we have created a platform called Gemini 1, and I will use that as an example. It's been created for COVID-19 patients, and it's based on reference digital twins. Now, the idea here is that when the patient uses the Gemini platform, he or she is being classified as a member of a group of digital twins. And then based on that, the medication is being suggested. In more detail, we are using publicly available data sets, single-cell RNA sequencing, in order to create reference digital twins, so-called proto-twins. We make digital twins for all of the subjects in the study. We make drug predictions for all of those patients, and we group the patients based on how similar they are in the sense of what drugs would be recommended for those patients. So we can find a uniform drug list that is suitable for all group members. And we can also identify biomarkers that would separate the groups of patients.
Sorry, Danuta, you have 5 more minutes.
Thank you. So we can identify protein biomarkers that can help us to separate the patients into main groups. And now when the patient seeks medical attention, the clinician may ask for measurement of those specific biomarkers, input them into the platform, then the platform makes the classification. So assigns the patient to one of the groups for which we already have drug recommendations, which are then returned to the doctor. The doctor reviews the recommendations, discusses the output and next steps and the treatment with the patient, decides on the medicine, and then cures the patient, hopefully. This is an example of the real life patients. So those are two subjects from a study that we have meta-analyzed in the process of creating the platform. Those are two representatives of the same sex. They have very similar age. They were both severely ill with COVID-19. The main difference between those two subjects are the comorbidities, so other diseases those patients had. Patient A, according to our algorithms, was assigned to group 2, and the drugs that this person was administered were very lowly ranked, which means that we don't predict that those drugs would be helpful for that particular patient. Patient 2 has been administered the same medicine, but those drugs were ranked much higher on the list, especially ribavirin. And we predict that those drugs might be more efficient to treat that patient, even though in both cases, we would not recommend those medicines to treat any of those patients because those medicines were fairly low on the rank. So we would recommend other medication for them. But in real life, they were treated with those medicines. So for patient A, we predict that those drugs wouldn't help. For patient B, we predict that ribavirin might have been helpful. In fact, patient A sadly deceased, even though he had only one comorbidity, whereas patient B was discharged, regardless of multiple comorbidities. So this is the platform we are currently working on. The platform is already in a state that can be possibly used, but we are still working on it, adding more data and refining the cutoffs and calibrating the parameters for the classifier. But I think that it will be revolutionary for the healthcare system.
Danuta, you have 2 minutes.
Yes, I'm almost done. So this is what we do now. We are using reference digital twins in order to recommend medication. But in 5 years, we plan to expand in order to invite people to create their own digital twins of healthy individuals so that we can follow a person from health to disease, prevent and diagnose as early as possible, hopefully before the symptoms occur, and that we can recommend the treatment based on the patient's own healthy state reference. So we don't compare patient to a population, but we can use patient's own health state as a reference in order to identify mechanisms and those disease genes that we would like to reverse to the healthy state. Now, there are very many challenges here. Many of them are linked to the big data analysis, data privacy, protection of the patient's biological information. And most of those challenges are linked to big data analysis and other analysis of the clinical data we know so far. So there are some solutions that we can reuse and experiences that we can reuse to overcome those challenges. So in summary, I think that digital twins are the future for personalized medicine. Thank you very much for the attention.
Thank you, Danuta. Excellent presentation. Now we have come to the Q&A session. There are 2 questions here that could be answered by both. I would read the first one. it's time for the speakers to take questions from you. Okay. Thank you, Laili, for the reminder. So in the future, this is the first question, right? In the future, will the risk of kidney failure as currently happening in Indonesia be reduced, or will there be possibility to get worse than it is now? And will the trust to the doctors decrease? Can technology ever be more accurate than human diagnosis that has been studying for years? Prof. Hera, would you like to answer first, and then followed by Danuta, please? Thank you.
Ina.
Yes.
Yeah. The question is whether the risk of kidney failure could be reduced in the future. I think we have to understand what is the cause of this kidney failure. If the cause is due by the risk, which is already detected, that we could actually reduce or we could prevent. But I think the one kidney failure that happened in Indonesia or Gambia is something that has to be studied first. What is the cause? Whether it is really because of the drug itself. So, and will the trust to the doctors decrease? I don't think so. I don't think that, because doctor is the one who actually becoming the person that they will see and they will seek for help. So doctors also, including public, has to know the future of medicine, and they have to understand whether it will become one of the session that given during their study or in the clinic. I think that make us the trust to the doctors not decrease, because they know the latest technology, including digital twin. I think, can technology ever be more accurate than human diagnosis? Well, I think this is asking Danuta, but I think technology coming from human brain. So yes, the important thing is using technology, which is built with a quality assurance and quality control. We might have a result which is more stable. No human error. I think that's all. But otherwise, technology coming from human brain, in my opinion.
Okay, so Danuta, could you also comment on that first question, please?
Yes, my pleasure. So I totally agree. I think that the technology can help to decrease human error. We have to also take into account the technology bias. So if we train the technology on wrong data, then the output will be biased. This is why we cannot, I don't think we can exclude the human to review the output. It's crucial that the clinicians would have the saying in the end, which to review the output. But in certain cases, I think that the technology may drastically help for improving the diagnostics and treatment predictions. Thank you.
Okay, thank you both, Prof. Hera and Danuta. We will continue to the next questions. Pragmatic questions. How affordable, this is for Danuta, I think, and also probably for the genomic testing for Prof. Hera. How affordable the technology will be in the next 5 years? What's the prospect of people living in global south like Indonesia? Perhaps before we ask Danuta for the advanced technology, we would also like to ask Prof. Hera for the genomic sequencing availability in Indonesia.
Thank you very much, Ina. Okay, for the genomic, it was the latest price that we could have is actually about $1,000 per sequence. But
in
now,
Indonesia. Oh,
in
no,
Indonesia,
it's there.
no,
Okay.
overseas, but coming from Indonesia. But now it is about $200 already. So it's becoming lower and lower, very different when the first Human Genome Project was announced, $1 per base. So if the demand is high, then the price is down. Yeah. So maybe we could afford that.
Thank you, Professor and Danuta, for your digital twins technology. How far is it from being used by the Global South?
Well, the plan for the company at the moment is that we will gradually expand. So we plan to invite all countries all over the world eventually. And it's very, I'm the development person, so I'm not deciding on the price here, definitely no. But we want to invite as many countries and as many people to use the technology as possible.
Thank you, Danuta. So I hope that answered your questions. The third question is related, actually, I think it's related to prenatal testing. Will technology replace the emotional feelings that the human beings have? For example, there is a kind of DNA test. It's not a kind of DNA test. It's a prenatal diagnostic testing for pregnant women, whether their baby have autism, Down syndrome, etc. And if the mother decided to do abortion, won't technology be destroying the empathy of the human being? Professor, you are familiar with prenatal diagnostic testing and how genetic consultation could help that. Could you please answer that?
Yeah, thank you very much. I think the question that you asked is about the prenatal diagnostic. Prenatal diagnostic usually being asked from a couple who already have child diagnosed with genetic disorder. So for the next birth, if they would like to have another child, usually they will ask. They will go to a consultation first, because consultation is very, very important. If you actually have, say, for example, a positive result, so what will you do about it? Because the genetic counselor will actually give the advice, but will not suggest anything. So there are things that the couple has to discuss, both of them. Emotion or emotional feeling, it's not associated with the prenatal diagnostic test itself, because it is voluntary. It is not actually being
mandatory. It's not mandatory at all. It's
not mandatory. If the mother decided to do abortion, it's up to the mother. I think that's the question.
Thank you, Professor. I'd like to continue on that and ask Danuta, is there any digital twins application for pregnant women?
Not that I'm aware of at the moment.
So it's not being applied for pregnant women, right? So it's related to disease so far. Thank you. And the next question is that how far has the ethical discussions, Hera has mentioned about the ethical discussion about ethical aspects of the genomic examination about personalized medicine gone? Are there standards in Western countries that regulate personalized medicine service providers about how to manage the patient's data? It's related to the ethical aspects. Hera has already Hera, you have already mentioned briefly at the end of your presentation. Maybe you could elaborate a little bit on that.
Yes. Regarding that personalized medicine, so far we have not actually used that already, right? We are in the position of collecting data. And also Indonesia just started maybe next year to do the study on some diseases. But then still a similar question will be asked. What about service? This is about service providers. How to managing patient data. So it is very, very important that data of patient is should be confidential, only given to the patient, and if it is needed, then it will be anonymous. Unfortunately, I think with the service providers, I don't know where, in Western country, because it's already developed, some company who has done Ancestry Plus, and not only Ancestry, but also looking at the risk factor. So there are many regulations that has to be followed, not in Indonesia, in my opinion.
Okay, thank you, Hera. I think that question is more relevant to Hera, right? Danuta will answer the last two questions, and this is one. This one is for Hera as well. With the technology, how can we avoid the trappings of the desire to create the perfect human like the Nazis did it a century ago? So Danuta has already mentioned that there's no such things as digital twins for pregnant mother, etc. So it's more to the creating a perfectly genetically perfect human being. Could you, Hera, answer that, please?
So the technology for those kind of creation is not genomic, but it's CRISPR. It's genome editing, actually.
Genome editing.
That you try to get some of the DNA and replace it with a normal one. But it's kind of like a recombinant DNA past 25 years ago. Only that this is now, it's more simple and more, I think it is cheaper in terms of technology and easier. And you have to understand that with genetic study or any genetic experiment is the issue of ethical, legal, and LC is very, very strong in that. Although you might be able to, one baby is already actually the
CRISPR baby from China.
In China, using the human editing, but still the issue come again and again.
Yeah, so the regulation is very strict, right? It's tight and strict.
Yes.
So you cannot just create a baby like that. Okay, so I hope that answered your question. The last two questions is for Danuta. May we know how many samples are used in building Gemini platform? How many ethnic groups that did you use in training the platform itself? And can the platform be used as a prediction tool, for example, to predict whether or not the patient will suffer from cancer with the current medical data?
Excellent question. Thank you very much. So the Gemini platform is currently built upon not enough data. So we are expanding it. We are analyzing and collecting more data. Right now, it's only one ethnic group, but we are, as I'm saying, expanding that to include as much as possible. We are using publicly available data at the moment so that we can include just as many groups of patients, as many representatives as possible. And then we will move on to clinical studies and to validate that the platform is working. So when that's one part. There is always the problem of not enough data. We always want more, and to make the algorithms as unbiased as possible with the current technology.
It is possible as a prediction tool. The last part of the question.
Not at the moment. So Gemini is made to be a prediction tool, but for not diagnostics, but for the prediction of which medicine is going to be most efficient. In the future, we want to create new tool that would be used for a prediction whether or not the patient may suffer from the disease, and hopefully start the treatment before the disease happens or the symptoms appears.
Okay, thank you very much for the answer. I hope that answered your question, anonymous attendee. So the last one is from A. Tamin. What are the important factors in your methods and Gemini platform to improve precision personalized medicine?
Well, the main factor is that we use the patient's individual factors into the predictions. So we use, at the moment, we use the reference digital twins, but in the future, we would use models of the patient, him or herself. So the most important factor is that we don't use prejudices. We don't focus on things that could bias the result. We look inside the cells, what is happening there, what genes are being expressed, to what level, and how they change. I think that this is the most important factor here, that we can step away from assumptions that could lead us to wrong conclusions, but instead we look exactly what happens in that person, and then we decide on treatment or help to decide the treatment.
Okay, thank you. I'm going to ask the last question for this session. So what effects would precision medicine, this is for both speakers, what effects would precision medicine have to the notion of care, relationship between a clinician and a patient that is thought of already diminishing due to privatized practice of medicine, clinician-patient becoming producer-consumer? Probably Hera could start a little bit of discussion on this question. The relationship, okay.
Yeah, the relationship between
patients, yeah.
Who actually have privatized.
Yeah,
well,
whether you usually have this relationship with your doctor, right? A very close one. What about when personalized medicine is introduced? Will it change the care of the relationship? The care
I don't think so. It is becoming stronger, in my opinion, because you know and you understand the individual. The individual now is becoming, it's a kind like an intact individual. You know everything about him or her, and you will give them medicine as what is needed. The doses will be actually adjusted, maybe a consultation about lifestyle. So everything is actually given. I think this makes the patient is more trust is stronger between doctor and patient. I think that, yeah.
Thank you, Hera. And Danuta, could you add more on that or elaborate?
Yes, I think that it's exactly how I would see it too, that the trust in the doctor would be higher because the medicine would start to be more accurate. And I also think that this technology might allow us to reduce the time that the patient, or give the doctors more time to spend with the patient, because if the patient doesn't need to return over and over again to the doctor, then the doctor can focus more and spend more time with the patient in the room.
Okay, so thank you both speakers for your answers. Do we still have any questions? Burning questions? Because this is something new, especially for Indonesia, right? The personalized medicine, the digital twins, they are both something to be related to our future healthcare. So we need to know really well about this personalized medicine. Oh, we have a question here to Danuta. Sorry if I missed. Could you explain about the AI bias in the challenges of digital twin practices? The AI bias, yeah.
Yes, so the AI bias means that if we train our models, if we train our technology on the biased data, so let's say, as one of the listeners has mentioned, just a group, subgroups of patients on a specific population, on a specific condition, then the output of the output will be also biased. So we need to make sure that the tools we are using, we are training on the right population. For us, it means all possible available data, so that we don't have tools that are racist, for instance, because they are trained on a specific population. So yes, that's the main goal.
Okay, so I think if there is no more questions, last offer. Anybody wants to ask anything? Burning questions? We have two very prominent experts here, which is not easy to get them together in one session. Okay, no? Okay, if no more questions, I would like to thank both the speakers and the audience for joining this session. The next session, The Future of Food, will start in 30 minutes. It features Wong Kak Ming from Shock Meats, a company that produces seafood using cell technology. For this session, let's give a big applause to our two speakers and also for the audience. Thank you very much.
Thank you very much.
Thank you.
Thank you, Hera and Danuta. Bye.
All.